primary monocot root anatomy or primary monocot root TS. Primary means before 1 year the plant parts are called primary parts. Primary monocot root TS or transfer section shows this type of diagram. Monocot root TS shows mainly three major portions. One is epiblemma, second one is cortex, third one is stele. Epiblemma is also called epidermis, all piliferous layer. Piliferous layer or epiblemma or epidermis. Only in root anatomy, epidermis is also called epiblemma or piliferous layer, because from this epidermis some cells are produced root hairs. The root hairs are mainly helps in absorption of water, minerals. So the root hair function is absorption. The root hairs are developed from the epidermis, root epidermis. The root epidermis is also called absorption layer. Absorption layer is another name is epiblemma or piliferous layer. Below the epiblemma, cortex is present. Cortex mainly made with the normal parenchymatous tissue. Mainly the cortex helps in storage of starch food material. Below the cortex or at center region of monocot root, one siphon-like structure is present. The siphon-like structure is called stele. Stele surrounded by two major layers. The boundary layers are endodermis and pericycle. Endodermis is a outer layer of stele. It made with rectangular parenchyma cells. The endodermal cells are also called starch layer or starch sheet. The starch cells present in the endodermis, each endodermal cell covered with starch strips. The starch strips are also called Casparian strips. Casparian strips are first identified by Casparin scientists. So the strips are also called Casparian strips. And one more important point in the endodermis, opposite of the protoxylem, Pauses cells are present. The Pauses cells main function, that Pauses cells are allows water, minerals from cortex to stele. Below the endodermis, pericycle, it is single layer or multi-layer of pericycle is present. Pericycle made with rectangular parenchyma cells. Sometimes pericycle produce lateral roots. Below the pericycle, vascular tissues are present in the radial position. The vascular tissues are xylem and phloem. Phloem alternate with xylem. The xylem shows mainly two types of xylem cells. One is large size of xylem cell, one is small size of xylem cells. The large size of xylem cells are called metaxylem. Small size of xylem cells are called protoxylem. Based on the arrangement of these protoxylem, in monocot root TS, exarch protoxylem is present. Exarch protoxylem means protoxylem present towards cortex or protoxylem present towards epidermis or epiblemma. See, in this monocot root, more number of protoxylems are present towards outside. So this situation is also called polyexarch protoxylem. Many protoxylems are present towards outside. So this situation is called poly. Poly means many exarch protoxylem. Alternate of xylem, phloem is present, and its center region of stele, parenchymatous pith or medulla is present. In this parenchymatous pith or medulla, parenchymatous cells are compactly arranged without any intercellular spaces. It is monocot root anatomy. So monocot root cannot participate in the secondary growth. Monocot roots cannot participate in the secondary growth. Now next we discuss about the primary dicot root TS. Primary dicot root TS similar to the monocot root TS means it is also shows three major parts. Dicot root, primary dicot root. Primary dicot root shows three major parts. One is epiblemma, cortex and stele. Epiblemma is also called epidermis or piliferous layer because it mainly helps in absorption of water and minerals from the soil. The epiblemma produced hair-like structure they called root hairs. They are in unicellular structure. They are also called unicellular hairs. They are mainly helps in absorption of water and minerals from the soil. Beneath of these epiblemma, cortex is present. Young dicot root containing simple type of cortex. The cortex mainly made with parenchymatous cells, but it slightly developed into adult age. The cortex divided into outer cortex, middle cortex and inner cortex. If the outer cortex made with collenchyma, it helps in mechanical support. Middle cortex made with parenchyma, it helps in storage of starch food material. And inner cortex sometimes made with parenchyma, sometimes made with collenchyma. If they made with collenchyma, it gives mechanical support to root. If it made with parenchyma, it gives storage of starch food material to plant. Beneath of these cortex, stele is present at center region of dicot root. It look like a siphon-like structure. The siphon-like structure part is called stele. It's surrounded by two boundary of cells. One is endodermis, second one is pericycle. Endodermis is made with rectangular parenchyma cells. That all endodermal cells are filled with starch food material. So that endodermis is also called starch layer. The starch layer of cells covered with starch strips. The starch strips are first identified by Casparian scientists. So these strips are also called Casparian strips. And in this endodermis, opposite of the protoxylem, passage cells are present. The passage cells mainly helps in allowing of water, minerals from cortex to stele. Beneath of these endodermis, single or multi-layer of pericycle is present. It is made with rectangular parenchyma cells. In this dicot root, the pericycle mainly helps in production of lateral roots, and they are also helps in secondary growth during the development of pellogen like cambium tissues. Below the pericycle, vascular tissues are present in the radial position. Here it is in the dicot root, only little bit of vascular tissues are present. Only 4 xylem strips, 4 phloem strips are present in this stele. It is stable number in the dicot root of vascular tissues. The xylem shows mainly two types of xylems. One is large size of xylem cells are called metaxylem. Small xylem cells are called protoxylem. Protoxylem present towards outside. Towards outside means towards cortical region or towards epidermal or epiblemma region. So this situation is also called exarch protoxylem. In this dicot root, only 4 protoxylems present towards outside. 4 protoxylems is called tetra. Tetra protoxylem present towards outside. Tetra exarch protoxylem. Alternate of these xylem strips, phloem is present. Phloem main function translocation of organic solutes to total parts of plants. Xylem main function is conduction of water and minerals to total parts of plant. Its center region of dicot root, parenchymatous pith is present. It is also called medulla. It made with normal parenchymatous cells. The cells are compactly arranged. The pith or medullary cells are helps in storage of starch food material. It is the primary dicot root TS. The primary dicot root participated in the secondary growth during the development. During enter into the second year, third year, the primary dicot root involves in the secondary growth. The secondary growth is normal secondary growth. Here it is the vascular tissues present in the stele. They are arranged in the radial position. Initially, in primary root TS, tetra exarch protoxylem is present. Tetra exarch protoxylem. Alternate of these xylem cells, phloem tissues are present. During the secondary growth, cambium tissues are developed. Cambium is developed from the cortex. Cambium tissues are developed from the pericycle or from the medullary tissues. The cambium tissues developed between the phloem and xylem. Between the primary phloem and primary xylem, new cambium strip is developed. The cambium strip developed from the pericycle or from the medulla. If once cambium is formed, the cambium produced continuously to outside secondary phloem to inside secondary xylem. To inside secondary xylem continuously developed, to outside secondary phloem continuously developed. After few years, we observed in the stele only secondary phloem to outside, secondary xylem to inside. It looked like it. The secondary xylem is also called wood. It mainly helps in mechanical support of root. It is the secondary growth of dicot root. And the differences between the monocot and dicot roots, here it is the dicot, monocot root it is the dicot root. These are the two diagrams explained with the monocot root and dicot root. The similarities, some differences are here it is. The differences are pericycle, vascular tissues, conjunctive tissues, cambium and xylem. First we discuss about the difference, pericycle differences. Pericycle in dicot, it gives rise to lateral roots, pellogen and a part of vascular cambium. So presence of pericycle, pellogen, vascular cambium is developed in the dicot roots. But in monocot roots, it gives rise to lateral roots only. It cannot participate in this secondary growth because secondary growth is completely absent in the monocot. Rarely few plants are participated in this secondary growth, but majority of the plants, they didn't participate in the secondary growth, like aloe vera, yucca. These plants may be participated in the secondary growth. And vascular tissues, usually the limited number of vascular tissues are present. The vascular tissues like xylem and phloem strips, like limited number, like 4 xylem strips, 4 phloem strips. Sometimes 2 xylem strips, 2 phloem strips also present in the dicot root. But in monocot root, the vascular tissues usually in more number of xylem strips, more number of phloem strips are present. So xylem strips are in more number. So we called polyexarch protoxylem in the monocot roots. But in dicot, di or tetraexarch protoxylem. It is the limited number. Di or tetraexarch protoxylem present in the dicot root. And conjunctive tissues. Conjunctive tissues are mainly made with parenchymatous. The cells are differentiated into vascular cambium. See, conjunctive tissues means medullary tissues. Medullary tissues are made with parenchymatous tissue. They compulsory develop into, differentiated into vascular cambium. The vascular cambium is developed from the secondary growth in the dicot roots. And in monocot roots, the conjunctive tissue is made with sclerenchymatous tissue. Sometimes it made with parenchymatous, but it cannot differentiate it into vascular cambium because they cannot participate in this secondary growth. Cambium, it is the fourth point difference. It appears as a secondary meristem at the time of secondary growth. Mean cambium is developed by secondary growth. and in monocot it is altogether absent. Xylem usually tetraexarch protoxylem, sometimes bi or diexarch protoxylem. In monocot, polyexarch protoxylem. Poly means more number of protoxylems are present towards outside. See, we easily identified which is monocot, which is dicot based on the diagram. See, it is the monocot diagram. Label is given, but without label, we can easily identify which is monocot, which is dicot. See, based on the presence of protoxylem. See, protoxylems are is more number of protoxylems in only monocot towards outside. That situation is called polyexarch protoxylem. But in dicot, limited number of xylem strips. Majority of the dicot roots containing tetraexarch protoxylem. Sometimes di or biexarch protoxylems also present in this. It is the dicot root, monocot root, anatomy with dicot root secondary growth.